Hi, this is Mr. Titanium, i.e. Richard Wagner, your host, Richard Wagner. I am so happy to be here. Today's episode, we're going to discuss a Reddit article that was about an alien's biology. Allegedly, there was a molecular biologist that spilled the beans sometime after 2010, having worked on a top secret project for a defense contractor. So let me start off with that. From the late 2000s to the mid 2010s, I worked as a molecular biologist for a national security contractor in a program to study exobiospheric organisms, EBO. The aim of the program was to elucidate the genome and proteome basis of their organisms. Although the study of EBCs has been ongoing for decades in other programs, the new high-throughput DNA sequencing technologies of the late 90s unblocked stagnant research in this area. Since then, several breakthroughs led to significant advances in our understanding of the genome and proteome of these beings. What we've learned so far has enabled us to outline some very disconcerting perspectives about our place in this universe. All right, so I just want to stop right here. I'm not going to continue reading just because I want to focus on the genetic portion. And even in the genetic portion, I'm only going to focus on a small part just so that we can do some sort of very quick analysis. Does this hold up to scientific scrutiny? And just in terms of that, I want to be entirely straightforward. It does not. It's a great piece of science fiction, but no. And I'm not going to go through the entire thing in order to demonstrate this. We're only going to talk about some things because it would just take too much time. And I think you'd get bored of it, actually, to be entirely honest with you. In fact, you're probably turning, you're just like, I've heard enough. First, I'd like to discuss their genetics. Their genetics are like ours, based on DNA. This fact was very puzzling for me when I first learned about it. We imagine that beings from an alternative biosphere would have genetics based on a completely foreign biochemical system, and surprisingly, this is not the case. Several conclusions can be drawn from this surprising revelation. All right, so I just want to backtrack a little bit. Full disclosure about disclosure, I'm not unbiased whatsoever. I'm actually quite biased. I believe that we are interfacing, or maybe members of our civilization are interfacing with non-human intelligence. Now, I don't know if it's extraterrestrial, terrestrial, or interdimensional. I can't tell you, but certainly there's evidence out there. Now, I haven't personally seen an ET or personally experienced, that firsthand, but there are plenty of credible people. And I think we're on the verge of some of the truth potentially coming out. But this paper isn't it. The philosophical thought goes something like this. If you were to print out a whole bunch of letters, A through Z, and do that many times, and take those characters and put them in a bag, and then shake the bag, and then randomly go into that bag, take out a character, put it down, it's unlikely that you'd be able to create a series of characters that would create even a simple sentence, let alone a complex sentence, let alone a sentence that's a thought process that's a paragraph, or a process that's a book. So obviously there needs to be an intelligence that went into figuring out how to arrange those letters in the correct order to create a sentence, to create a paragraph, to create a story. So there had to be an intelligence, there had to be a process, there had to be something that enabled this to happen. All right. It couldn't have happened on its own. You're saying, well, back 4 billion years ago, you just had a bunch of chemicals. How could those chemicals have just randomly gotten together and created RNA and then subsequently DNA? How does that happen? And how does it randomly happen once on Earth and then in other biospheres? The probability of that is astronomical. That's how it appears if you think of it in that context. But that's the incorrect context. A better way of thinking about it is that what you have is a bag filled with characters. Those characters have very specific grooves that only allow them to fit with other characters. So they can only fit one way. There's only one way that they can fit together. Not only is there only one way that they can fit together, but these characters are attracted to one another. So they fit together a certain way and they're drawn to each other. Now, when you take a bag and you shake it up, it's no longer random. The characters are attracted. So when you pull things out, of course you're making sentences. You're making sentences because the conditions prior to which were not exactly random. And that's the case. That is exactly what happens in terms of evolution and life on this planet and life on other planets. It's not random. Molecules are attracted to each other in certain ways and formulate bonds in certain ways. And I believe that RNA is actually one of those molecules, like water, like carbon dioxide, like many other types of molecules that are ubiquitous in our universe, that exist everywhere. And I believe this is the case because they're attracted to one another. There's that property that we're talking about. It's no longer random. And as a result of that, I don't believe that the formation of life is random at all. I think that it happens in ways that are different, and then evolutionary pressures come in. But I believe that DNA itself may very well be the building block of any life form that lives in a biosphere that has conditions similar to those that exist on Earth. So anywhere where there are conditions that are similar to Earth, the same elements and perhaps even similar temperatures and so forth, are going to create DNA, RNA, and then DNA ultimately. So I don't think it's surprising at all that if life exists elsewhere, and I believe it does, that it's based upon, deoxyribose nucleic acid. I don't think that that's a stretch. And the reason why I'm mentioning that is because he's implying that it's a stretch. And I think that most biologists would say that doesn't sound so crazy to them. I think that they would say that the fact that DNA exists elsewhere in our universe and is ubiquitous, if we find out that that's the case, really demonstrates that there's a process that occurs after the formation of a planet, and that process happens over and over again, and it's a chemical process. All right, back to the paper. All right, it starts with several conclusions can be drawn from this surprising revelation. The one that immediately comes to mind is that our biosphere and theirs share a common ancestry. They're eukaryotes, which means their cells have nuclei containing genetic material, which suggests their biosphere would have been separated from ours sometime after the appearance of this type of organism. So if you're going to have complex multicellular organisms, you need to have the regulation that is conferred by a structure of some sort of envelope around the nuclear material. So this also is not surprising, and it's not surprising that this is the way in which life evolves, and that life could have evolved separately on a different planet somewhere else in a similar fashion. And the idea that two things can evolve separately but have similar functioning is also a very common thing that happens in biology on this planet. All you need to look at is the coevolution of eyesight. And the two examples that are typically used are that of the mollusks, in particular the octopus and the squid, their eyes compared to the eyes of, say, the mammal. And it turns out that the evolution happened completely separately, and they are completely separate genes, and the morphology of the eyes are actually different. And in some ways, we could look at the mollusk, the octopus or the squid's eye, and say it's superior because of the way in which the retinal nerve and the retina exit the eye itself. But we have all these analogous structures, but yet the genes are completely and utterly different, and they evolved separately. So that means you can have similar structures that evolve separately. But in the same way that you can have similar structures that evolve separately, it's not hard to imagine that on different planets, you can have single cell organisms that develop, a nuclear envelope. That's not out of the question either. So these things aren't shocking. They're not surprising. They're actually, from what I'm thinking, they're somewhat expected. I wouldn't expect it to be any other way. I would expect that if it was different than that, for it to be shocking. The term exobiospheric organism is actually a misnomer, as it's a historical term. It's still used. Their genetics are not only based on the same genetic system, but they're also compatible with our own cellular machinery. All right, so once again, DNA, different organisms' ability to replicate DNA, and that DNA machinery for replication can be used in other organisms is not something that should be shocking or bizarre to anybody. Just think about a virus. Viruses are interesting. All right, so one of the criteria for life, or one of the ways we try to define life, is one of the characteristics of life is the ability to replicate. It's an imperative characteristic. Without the ability to replicate, life isn't going to go too far. You know what I'm saying? But here comes along a virus. A virus can't replicate. It's literally like a cell wall surrounded by genetic material. All right. It's a capsid surrounded by genetic material. That's what it is. So this virus, when it infects a host, uses the host's cellular machinery for replicating itself. So here you have something that exists solely because the host's genetic machinery can work to replicate it. So we know that this is the case. This is, in other words, the person didn't even have to dig too hard to figure this out. And this isn't something that's remarkable. In fact, we customarily need to expand DNA all the time. But in order to expand DNA, we first have to separate the two strands. They're double-stranded and stuck together. They're stuck together with hydrogen bonds. And the way that you liberate the hydrogen bonds is through heat. And heat enables you to separate the DNA, thereby exposing the base pairs, so that you can have the DNA polymerase be able to replicate or transcribe. But in this case, we're thinking of replication. The problem is that the machinery to replicate the DNA polymerase gets denatured by heat. We all are familiar with eggs. And when you crack an egg and put it into a frying pan, all of a sudden it goes from being see-through to being white. What's happening there is that the egg album, which is really protein, is becoming denatured by the heat. And instead of the protein being nicely folded, it turns into nice long strings. And in that process goes from see-through to white. That's called denaturization. But that denaturization happens to DNA polymerase. So the heat that we need to apply in order to expose the DNA substrate also destroys the DNA's polymerase. So that left us with a problem. But we wind up having organisms on this planet that live at the bottom of the ocean next to thermal vents, where the heat is actually above the boiling point of water. And at these temperatures, the cells are dividing, they're replicating, cell replication occurs. Therefore, their particular DNA polymerase is resistant to heat. Well, we use that heat-resistant DNA polymerase called Taq polymerase, and we used it to expand our DNA. So now we can heat the DNA. We can separate the hydrogen bonds, and we can use the Taq polymerase for expansion. But now we're using a different organism's cellular mechanism for the expansion of perhaps human DNA or any type of species DNA. So the idea that some other organisms' cellular replication machinery, when it comes to DNA, works on ours is quite truthfully expected, not shocking. All right, so let's continue onwards. This means that you can take a human gene and insert it into an EBO cell, and that gene will be translated into protein. And this, of course, works in reverse, with a human gene inserted into an EBO cell. Okay, so this also is not something that's at all peculiar. We do this all the time in molecular biology. We create transgenic organisms in order to, you're not going to go ahead and muck with a person and change their genes, because that just is completely unethical. So we use model organisms and muck with their genes. But when we do, we push genes in or take genes out. And I'll give you a perfect example of that. There's a gene called FOXP2. Now, this gene is associated or believed to have given Homo sapiens their ability to speak. It's actually, that gene existed in the Neanderthal, but there's there's differences in that gene that we believe led towards us developing speeches where the Neanderthal didn't. So we took this FOXP2 gene and inserted it in, of all places, of course, where else would you insert it? In a mouse. And no, the mouse did not start speaking. However, interestingly enough, the mouse was able to complete maze problems far more quickly than the non-transgenic counterparts. So we've been putting genes in things all the time. That's just something that we do. So this is not something that's at all surprising, and it's not at all surprising that these genes can be translated and that proteins can be created. It's not, in other words, it's part of the everyday normal aspects. So if you're writing a science fiction, like if you were Gene Roddenberry, what he did is he used a lot of scientific stuff in order to, make something seem realistic. And that's kind of what this feels like. There are important differences in post-translational modifications that will make the final proteins non-functional, but I discuss these later. Their genome consists of 16 circular chromosomes. Okay, so I just got to address 2 points. With regard to post-translational modifications, wow, that is a mouthful, but let me just at least describe what this is. Whenever we talk about proteins, we talk about them really we describe them in 4 tiers. We have their primary structure, secondary structure, tertiary structure, and quaternary structure. Their primary structure really discusses the individual components that make up the protein, the individual amino acids, and how those amino acids are strung together. That's the primary structure. Now, how those things are strung together lead towards secondary structures. Secondary structures are the different types of folds that are made. And towards those ends, there are a variety of very, well-known motifs. For example, a beta barrel or an alpha helix. These are different types of motifs that have been discovered and known about. The tertiary structure are how these different folding things kind of bend together. And right at the tertiary structure point, we have a lot of proteins that are functional, that we use, that don't need any further modification or anything. It's just that there are others that are a little bit more complex, and those get held together with something known as disulfide bonds. Disulfide bonds, the existence of the disulfide bonds brings us from brings us to our quaternary structure. So, and an example, incidentally, of a disulfide bond occurs in hair. So when hair is very curly, there are a lot of disulfide bonds. So when you get a perm and you smell sulfur, that's because they're adding it to your hair. They're adding disulfide bonds to make your hair curly. That's how that works. So the idea that there are post-translational differences, there are post-translational differences in proteins amongst different people, let alone amongst different species. This, again, is not at all surprising. The genome consists of 16 circular chromosomes. No, can't happen. All right. So what the author is trying to do is these creatures apparently, he's trying to make them out like they live forever or they live for a very, very long time. Now, the problem with mortality is the problem of telomeres. Every single time a cell reproduces, at least with the type of cellular machinery that we have here on this planet, we shorten our telomeres. Every replication means that the telomere just gets a little bit shorter. And eventually you get to a point where the telomere is so short that the cell dies. And obviously, the length of a telomere is indicative of lifespan. So if your telomeres are very long, you're probably young. And if you're like me and you have teeny weeny telomeres, you're probably getting a tad bit old. So telomere length has a lot to do with aging. And so what he's doing is that he's circumventing the problem of telomere length by suggesting that their telomeres are circular. And if their not telomeres are circular, their chromosomes are circular. And if their chromosomes are circular, then there are no telomeres at the end to get shorter. So you wind up resolving that issue. But in resolving the issue of decreasing the length of telomeres, he's negating the real benefit and functionality towards having chromosomes that are linear, and that comes in the form of regulation once again. And what you have to understand are that for one thing, I want you just to think about your own body for a moment. Inside your body, every single cell has the identical DNA. But look at how much different a heart muscle is from a neuron. They're completely morphologically and functionally disparate. Well, how does that happen? That happens due to the way in which the cells regulate themselves, due to the protein products that are being produced. Well, a cell that is a heart cell produces, characteristics that are good for that. How does it do it? By turning on and off genes. And there is we can speak forever on how we regulate that process. It's actually quite interesting. But one of the ways, as an example, is through the geometry of the chromosomes. So, for example, there are areas. So, when you go to transcribe a gene, the machinery has to be seeded onto the DNA. The first thing that needs to happen is you need to actually find a place where you can open the DNA up. All right, because the DNA is shut, and you have to get to the substrate. But once you get to the substrate, you have to read what's at that substrate. You have to read what's at that substrate. But in order to start reading what's at that substrate, you have to attract other things called translation factors and other things. And in order for the machinery to work correctly, there has to be portions of the DNA brought over that are called enhancers. But it turns out that the enhancer for a gene doesn't need to be on the same strand. You can get an enhancer from a different strand of DNA that would enable the RNA polymerase to do the transcription, or rather translation, to be more accurate. So that translation can occur from another chromosome's enhancer. So in this way, the geometries of a linear chromosome and the way that they can bend and interact with each other are imperative for the complexities needed with a multicellular organism. A simple multicellular organism, like a flatworm. A flatworm requires so much regulation that it negates the simplicity of a round chromosome that prevents you from having those complex geometries that can only occur through large strings of linear nucleotides. So while it may sound elegant in terms of cellular aging to have circular chromosomes, from the standpoint of regulation and from the standpoint of complexity, you cannot have the complexity necessary to support a simple multicellular organism, let alone one that travels through the galaxy. All right. So I guess to some degree, I'm going to end the discussion with this. This was an interesting read. And I guess some of the problems that I have a little bit come down to the fact that in this day and age, it's getting harder to know what's real, what's not real. We know that there are cover-ups going on. And this type of information, it doesn't move our agenda along very far. You know, one of the things is that even in writing this, the thing that occurred to me was that all those things that he found unusual, like, wow, it's like DNA, and I didn't think from a different biosphere. I didn't think biochemically it'd be from a different biosphere. If the author just thought a little bit harder, what he could have said, and then this would have given him substantially more credibility if he really wanted to pull a lark on people. You have to understand that every amino acid is coded by 3 codons. 3 codons are 3 DNA nucleotides. So DNA you can think of as a lettering system. A for adenine, T for thymine, G for guanine, and C for cyanine. So cyanine, guanine, thymine, and adenine. DNA is basically, the alphabet of DNA is made up of 4 letters, A, T, G, and C. These 4 letters can be combined in groups of 3 to make up amino acids. For example, the amino acid for methionine is AUG. The amino acid for proline is CCC. So different codes, different 3-letter codes represent different amino acids. Now, there are 20 amino acids. However, if you think about it, if you only had one codon, that could only be 4 possibilities. 2 codons is 4 possibilities times 4 possibilities, makes 16 possibilities. And with 3, which is what we have, it's 64 possibilities. So those 64 possible different codons get divided up into 20 different, get coded to 20 different types of amino acids, as you can see. But it turns out that in our own bodies, this is not consistent. A long, long time ago, when in the early part, when eukaryotes were just beginning to form, they developed a relationship with a certain type of bacteria. We now call that bacteria the mitochondria. And that mitochondria's DNA is actual codes for amino acids differently than our nuclear DNA, just a little bit. So if that can happen within our own body, then in different biospheres, the coding systems have to be completely different. So if the person wanted to write something that was more convincing, to suggest that we shared a similar evolutionary ancestor, which was what the person was trying to indicate, but I think failed at that task, what they could have said was, my God, we even use the same amino acid codon system. And if our codon charts match the EBOs, then I would say that, in fact, we had to have had a similar ancestor at some point. So that, but he didn't say that, all right? And that's what leads, that's just additional evidence that this was a lark produced by a person that had some knowledge, but was certainly not PhD level. It's certainly not what this is. So, but he was very creative, and I enjoyed reading it. Listen, have a great evening. Thank you for spending this time with me. Oh, please remember to like and subscribe. You must like and subscribe. Without that, this channel cannot go forward. Thank you.